Today, the topic of our discussion is structure of DNA. DNA means deoxyribonucleic acid. Just imagine that you are standing in a street and looking toward a stair moving upward in spiral shape, and the step of the stair are made up of rods. One step contains three rods, then the next contains two rods, then the next contains three, then two means the formation of the steps contains two rod, three rod move in an alternate fashions. And if I talk about the structure of the DNA, the structure of the DNA is the same as the DNA contains two strands in the form of double helix. One strand is this and the other is this one. And if I talk about the backbone, the composition of these two strands are made up of a chain of two deoxyribose phosphate molecule. And if I talk about how these molecules are bonded to each other, there is present phosphodiester bond between these two deoxyribose phosphate molecule. And listen carefully that this is carbon number 1, 2 and 3. The carbon number 3 of one molecule form a bond with 1, 2, 3, 4 and this is fifth carbon of the other molecule, and phosphodiester bond is present. And third point in is that the strands are anti-parallel to each other. It means that both these strands progress in an opposite manners. The starting point of one strand will face the ending point of the other strand, and the strand start from 5 and end at 3 end. And this is carbon number 1 of the molecule deoxyribose, and this is also carbon number 1. This carbon number 1 form a bond with two structures, either with purine or with pyrimidine. In DNA, purine and pyrimidine are of two types each. Purine are adenine and guanine, while pyrimidine are thiamine and cytosine. Yeah. Next point is that base purine. These both two strands are held together through these two dotted line that are structures, that is a hydrogen bond. And hydrogen bond formed between purine and pyrimidine. There are present two hydrogen bond between adenine and thiamine. In DNA, always adenine form bond with thiamine and guanine bond forms bond with cytosine, and three bonds three hydrogen bonds are present between guanine and cytosine. And in one DNA molecule, thousands of base pair are present. And now again imagine that structure of the stairs. If I ask you to break the steps of the stair, you should have to consume more energy in order to break the step made up of a three rods than the step made up of a two rod. So, in the same manners, if I asked for DNA denaturation, the DNA contain a high proportion of cytosine and guanine will need it high melting point or high melting temperature to convert the double strand into a single strand. And in physiology or in medicine, in 2009, a Nobel Prize is awarded for the discovery of that how chromosome is protected with these two things, telomeres and telomerase. So remember one thing, that in biology enzymes name contain ASE at their end, like telomerase, dipeptase, transcriptase, peptidase. These are all enzymes. So if I move toward the telomeres, telomeres are a cap at the end of a DNA helix in the form of eglets, and eglets are the ending cap of the laces, and it means that no further progression. And in vertebrates, telomeres are a repeat of a sequence like thiamine, thiamine, adenine, guanine, guanine, and then guanine. This is a sequence of a telomeres that attract the proteins that form the protective cap at the end of a DNA strand. And if I talk about the formation of the telomeres, they are made by telomerase. And as you know that all enzymes are protein in nature, so as telomerase is enzyme, it is also made up of a protein in addition to RNA. But the function of RNA is that it contains the information in order to form the telomeres. And in the second step, in aging cells and in cancerous cells, telomerase reduce in numbers. The reduction of the telomerase results in the chromosomal instability. If the chromosomal instability occur in a cell, that cell become abnormal, that causes cellular damage. And if I talk about how many base pairs are present in DNA per turn of a helix, so 10 base pairs are present per turn of a helix. That's all about the structure of a DNA. Thank you.